let's imagine this is a cell. Can we do that? And this is going to be a cell where 2n equals 2. So what does that mean? How many pairs of homologous chromosomes are there? One pair. That's it. All right. So I'm going to represent that one pair as an open chromosome and a closed chromosome. So one that's colored in and one that's not colored in. So how similar are these two to each other? 99.9, like ivory soap. So, sorry, that's a reference. No, you guys even get that? You know what? On ivory soap it says 99.44% pure. You guys see that? Okay. So the first thing we have to do whenever we're going to divide a cell is we have to copy the genetic information. And that occurs also in meiosis. So first thing we're going to do is we're going to copy the DNA. And when we copy it, we're going to make our sister chromatids. Sister chromatids, 100%. Sister chromatids, 100%. Homologous chromosomes, 99.9%. Okay. Now, if this were meiosis, what would happen? They would line up and then split apart. But in meiosis, this weird thing happens where they form this structure. It's called a tetrad. And tetrad refers to 4 parts coming together in one. And it's actually all 4 double helices of a given chromosome type all coming together into one sort of unit. So when you sketch it with our simple drawing here, our cartoon sketch, you'd have to draw the tetrad looking like this. And if you were to think about it on the DNA double helix level, I don't know if this is actually going to work, but I'm going to try it. It would look like this. These are identical, these are identical, these are homologous. So if you really looked at it like it really looks, they're not lined up next to each other like little, I don't know, sticks. They're all sort of intertwined around each other. And what happens when they're intertwined around each other is they do this weird recombination where one homologous chromosome literally recombines DNA, like the molecule itself, not just information, but the molecule itself with its neighbor. So if you imagine, and I'm just going to sketch a single recombination event because it just gets confusing to draw if I sketch more than that. But imagine along the length of this chromosome, 5 events occurring where you're swapping a piece of mom's DNA onto dad's chromosome and vice versa. So what it would look like after a recombination has happened is this. And if you were to sketch it at the DNA double helix level, it would look something like this. on purpose. Maybe I was. Okay. So I hope what you can see, and I think you can even see, even though this sketch is a little bit, it's not so great, is that I've moved part of dad's chromosome over onto mom's and vice versa. Okay. And how that occurs is kind of incredible, and you're welcome. We're not going to talk about it, but you're welcome to look into it in more detail if you are interested. It's complicated. There's lots of proteins involved in the process of recombination. It's a very ancient process. And you're welcome to learn more about it. We just don't have time to talk about it in this class. Okay. But it's a cool process. And it requires sort of, it's kind of like a, if you're good at like Tetris, you're probably good at understanding how recombination works. So the next thing that occurs is we're going to move on to something that's very similar to what we've seen before. This tetrad is going to line up in the center of the cell in metaphase, following recombination. So it'll look, if you imagine this cell with this dotted line down the center, the tetrad is going to line itself up at that center point like you saw in the meiosis slides you just looked at. Switch pens, guys. Now, in mitosis, what we did is we pulled on the two sister chromatids, and the sister chromatids dissociate from each other and move towards the poles of the cell. Right? In this case, what we do is we pull on the pair of sister chromatids, and they separate from the other pair of sister chromatids. All right? So what happens is this pair of sister chromatids moves together to one side. This pair of sister chromatids moves together to the other side. So you'd end up with a cell that looks like this. I should have drawn them in action. Sorry. I'll redo that. I don't want it to deprive you guys. Then we move to a situation where we have our two pairs of sister chromatids on either side of the cell, and the cell beginning the process of actually dividing itself. So if you went through and named these phases according to sort of the way we described mitosis occurring, you'd say that this stage here, and for us, this is just fine. Prophase describes the point when those tetrads are together. Metaphase describes when they're lined up in the center of the cell. Anaphase, when they're separating from each other. And telophase, at the conclusion, when those pairs are at opposite sides of the cell and the cell is beginning to divide.